Good afternoon, one and all. I'm very proud of being a part of IHRD, and I'm very much proud enough to part of this conclave. I thank all the coordinators for giving such an opportunity to present one of the application of generative AI in human chromosome analysis. I am including in this presentation my research finding. I was actually a research scholar at College of Engineering, Karunapalli. Currently, I am working as an assistant professor in College of Engineering, Attingal. And my research was guided by Dr. Gopakumar C. I have already submitted the thesis. As we all heard about from the previous sessions, generative AI refers to the subset of artificial intelligence which is generating contents. So the content may be text or maybe image or maybe video, audio, anything. So far we have discussed about how ChatGPT is generating text. And the heart of generative AI is basically the generative models. We have seen transformers, generative predictive transformers, that is really the heart of ChatGPT. Whereas we can use other generative models like variational autoencoders and generative adversarial networks. Basically, generative adversarial networks are used in image processing and generating image content. In medical research, we must have a lot of data, otherwise we can't give better results. So for data augmentation, initially generative adversarial networks are used. But certain type of generative adversarial networks are there. They are basically called as conditional generative adversarial networks. One among them is pix-to-pix GAN. So which is a conditional GAN that is basically used for image-to-image translation. So in my work, pix-to-pix GAN is extended for medical image segmentation. Especially I have used this technique for semantic segmentation. And this is a work on automated karyotyping system. We all know there is a field called cytogenetics. Cytogenetics, there is a tool called automated karyotyping system. In automated karyotyping system, basically we are taking the input of G-banded metaphase image that is shown in the left side. So that is the G-banded metaphase image in which the chromosomes are clearly visible. What we have to do is we have to pair and classify all the 23 pairs of chromosome into a karyogram. At that time, the cytogenetist can analyze and visualize what are the different disorders existing in this karyogram. There are basically two types of abnormalities, disorders. One is numerical abnormality and another one is the structural abnormality. There are certain examples for numerical abnormality. One is a trisomy 21. As you can see in the left figure, there is 3 chromosomes in 21. That is a genetic disorder which causes Down syndrome. Another abnormality in a numerical sense is that Turner syndrome, in which actually a Y chromosome is missing for a male. And in Klinefelter syndrome, more X is there for a male. So all these are the genetic syndromes in a numerical sense, whereas there are certain structural abbreviations will also occur. One example is Philadelphia chromosome. That is the main cause of chronic myeloid leukemia. So this structural changes that 22 chromosomes, in one chromosome, some portion is deleted and is appended in a 9 chromosome. That translocation is basically called as 922 translocation and is causing chronic myeloid leukemia. So in RCC, all these type of, RCC as well as Child Development Center Medical College, all these type of research is going on. The main problem in automating this karyotyping system is overlapping and touching chromosome. In this figure, you can see when we are having a metaphase image, there may be a lot of touching chromosome and overlapping chromosome. So what they are doing basically is they are pseudo coloring all the touching and overlapping chromosome. That will take days and they will rearrange all the chromosomes separately, and they are doing the karyotyping. It will take a lot of months to generate karyograms, and after that they are visualizing whether there is any abnormalities or not. Surely the patient's results will be very much delayed. So in that scenario, we can make use of this automated karyotyping system. If we are having a system that is enough to automatically segment all these type of touching and overlapping chromosomes, the problem will be easier. But the problem is very much complex, as illustrated in the second figure. The morphology of the overlapping and touching may be changing, and the number of chromosomes participating in overlapping or touching may also vary. So that is very important problem. That is again an open issue. Anyone who is interested in doing research in this field, you can. So basically, we have identified that there is scope of using this pix-to-pix generative adversarial networks, but the problem is data. We are having not much data published. Anyway, we use this pix-to-pix GAN, and basically there are two types of CNNs. One is generator and another one is the discriminator. Generator is actually generating the fake samples, and discriminator is actually distinguishing whether the fake sample matches or whether it is a fake sample or not. So until the generator defeat the discriminator. So at that time the discriminator cannot identify whether there is whether that is a real sample or a fake sample. So ultimately, we can see UNet. UNet is a wonderful architecture for semantic segmentation, and it is an encoder-decoder type architecture, and even with small amount of data, it can work well. So basically, pix-to-pix is using generator as the UNet architecture. In our method, we have modified this UNet architecture. I'm not going into the details because of the time limitation. This is the encoder and the expanded decoder blocks, and this is the main architecture in which you can see the number of parameters, input shape and output shape, etc. And there are discriminator, which is a fully connected network, as you can see here, and these are the details of that discriminator block. And one novelty we have introduced in this area is there are a large number of loss parameters. The adversarial learning, through the adversarial learning, we can optimize the loss. That is, the adversarial loss is there. What we have to do is we have to minimize the loss. That we have already heard from previous sessions. We have introduced a combination of three loss here. One is the binary cross entropy with logic loss and Turkey loss. This Turkey loss is very good at handling the class imbalance problem. That is why we have introduced there along with the L1 loss. And this is optimized through the training of 250 epochs as well as 500 epochs for both the databases. One database is publicly available, that is a FISH data set. All these are the sample you can see, and the labeled images is also available. And but they are actually the FISH data set. When you go through the cytogenetic imaging techniques, there is a technique called FISH. But in RCC and CDC, they are basically using the G-banded images. That is not at all available. So we have generated at RCC through a cytogenetic procedure, and we have already published the data set in the Mendeley data, and such samples are generated through our investigation. And we have learned the network both with the data set 1 and with the data set 2. And we have arrived at these solutions. I mean, we have tried for many combinations of loss function, and we have proved that our model with the proposed loss function is working well with good results. As well as we have compared with the, we have experimented on the FISH data as well, and we have got a more or less same result for the FISH data set as well. And we have compared for the other set of model in which we have used certain CGAN models with different type of generators, UNet, SegNet, nested UNet, R2 UNet, attention UNet, etc. And we have proved that our model works well. These are the qualitative analysis we have performed on RCC data, and we have proved qualitatively that our model is working well. These are the results on the FISH data. So with the time constraint, I am concluding. The presence of overlapping and touching chromosome in metaphase images is challenging open task on automatic karyotyping. We have identified that the generative adversarial model pix-to-pix can solve this problem to a certain extent, but the problem is availability of data set. If we are sure enough to generate large number of data for solving such problems, we can make use of generative adversarial network. Surely for novelty, certain thing we have to include, we have to ensure. With this, I am stopping. Thank you. Thank you for the opportunity.